Oh, we are already live there, right?
Okay, I think it just went up live on YouTube. I'm honestly amazed that it's so seamless, right? I'm using a new Zoom account, but it just lets me stream to the old
That's awesome.
YouTube channel without having to do anything, which I'm really grateful for because I have no technical abilities that would have allowed that to work well if there was a problem.
I'm grateful you do it because I don't have to. I will have to figure it out how to do it.
You know, Becky and Adam and I, in the very beginning, we sort of set everything up and we figured out how to do it. But if something breaks, I have no idea how to get it back, you know? So I'm glad it's been working seamlessly. So I think we'll just wait another maybe minute and a half.
I did put it up on Twitter again saying half an hour.
Okay. Fantastic.
It's nice still getting
see so many. Sorry, I was just saying it's nice to see so many new names, and also lots of you guys have been coming since the beginning. So friendly, maybe not faces, but friendly black boxes names. At least. It's wild that we're almost going to be at 2 years. So once we finish this session, we'll have 2 full years of this, which is
that's amazing.
It's like, it's depressing because it really hammers how long things have been strange and bad in the world. But I think it's wonderful that people are still excited about this and coming together. So
I think we still have a lot of people to, who could potentially present. And I mean, this made me realize how big the community is. Like cell migration itself is so huge.
Yeah, anyone in the audience, we're always, send abstracts at any time. Please, we probably won't look at them until the summer, but send them at any point.
Yeah, the earlier the better, because when we start organizing, it's always like, oh, now we have to look for the abstracts and put up advertisement and stuff like that.
No, we're very organized, very professional. And I'll let you take it away. Okay, when you're ready.
Welcome everyone for Cell Migration Seminars of 2022, and a very happy new year since we didn't meet in Jan. We have an excellent lineup of speakers for the series as well. I think Jen will put it up at some point, maybe by the end of it. I'm Ankita Jha. For people who are new, I'm currently a postdoc in Dr. Claire Waterman's lab at NIH in the United States. And I'm one of the organizers and moderators along with Dr. Jennifer Mitchell from Wesleyan University. She's a new PI, so yay. Today we have 2 wonderful early career researchers to begin the session and the series. We will begin with Dr. Shailaja Seetharaman, who is currently a postdoc in Professor Margaret Gardell's lab at James Franck Institute, University of Chicago. She received her PhD in cell and developmental biology from Institut Pasteur, Paris, working with Professor Sandrine Etienne-Manuel. She's interested in deciphering the cytoskeletal crosstalk mechanisms that contribute to cell adhesion, migration, and mechanotransduction in normal tissues, all our favorite topics, and how alteration in these cellular processes can drive disease progression. Today, I think she's going to tell us about her PhD work, which is going to be microtubule-actin crosstalk and mechanotransduction. Before I hand over the virtual stage to Shailaja, just some basic rules, not really rules we follow, is please mute yourself when the speaker is giving the talk. You can put up your questions in the chat. We really, really encourage that. And most of the questions we take up after the speaker is done. Unless it's a really, really pressing question and you want to clarify right away, you can unmute yourself. And over to you, Shailaja. Thank you so much for accepting our invitation.
Thanks, Ankita and Jen, for inviting me and giving me the opportunity to present at this really amazing seminar series. So I've been watching this right from the beginning, and it's been great, especially during the pandemic and the lockdown. All right, so my name is Shailaja Seetharaman, and I'm going to be talking a bit about my graduate school work that I did in Sandrine Etienne Manville's lab at the Pasteur Institute in Paris on the role of microtubule-actin crosstalk in mechanotransduction. So I don't have to tell this audience about cell migration, but I'm still going to, and here's my obligatory slide. Cell migration is important for various cellular processes during morphogenesis and development, during wound healing and tissue repair, immune responses, as well as contributes to pathological conditions such as cancer cell invasion and cardiovascular diseases. So I'm going to be speaking about cell migration of astrocytes. Astrocytes are major glial cells, and these are cells that are star-shaped, as the name indicates. They interact with neurons and other glial cells to control overall cerebral homeostasis. Astrocytes can also give rise to highly invasive brain tumors and astrocytomas, and therefore their functioning and normal functioning is very important in the brain. Astrocytes upon inflammation and injury can migrate, but as astrocytes are in the brain in normal physiological conditions, they are immobile, and only upon inflammation and injury do they migrate. So during inflammation and injury, they migrate towards the site of injury to facilitate tissue repair. In order to simulate this in vitro, we have a scratch wound migration assay with astrocytes plated in a monolayer, scratch down the middle where you see astrocytes migrate towards the gap to close and facilitate wound healing. So this is mainly the model system that we're using in the lab, and this is what I'm going to be talking about. As these cells migrate, they sense the extracellular environment through integrin-mediated focal adhesions. This then sends signals back into the cell to alter signaling pathways, gene expression, and cytoskeletal regulation. In particular, the actin-myosin network generates majority of the forces in response to these signals, which are then transmitted through focal adhesions onto the extracellular environment, completing this feedback loop termed as mechanotransduction. And this is a very important process in cell migration, proliferation, differentiation, etc. A lot of work has been done. A lot of work in mechanotransduction has been done in the context with mostly studying actin-myosin networks. However, very little is known about microtubules in this context. And during cell migration, microtubules grow and extend towards the cell front. As you can see here, EB3 plus tips of microtubules grow and extend towards the leading edge of the cells, where you can see focal adhesions marked in red. And microtubules not just come in close proximity to these focal adhesions, but also regulate their turnover in different steps like formation, maturation, and disassembly through exocytosis and endocytosis pathways, as well as a close crosstalk with the actin-myosin cytoskeleton to control contractility. There are several ways by which microtubules can be regulated, and one such mechanism is through post-translational modifications of microtubules. And in this talk, I'm going to be focusing on one such post-translational modification, which is acetylation. This occurs in the lysine 40 residue of alpha-tubulin and is the only post-translational modification that occurs within the lumen of microtubules. And acetylation is known to mark stable and long-lived microtubules, along with the other post-translational modifications such as deterioration. All right, so acetylation is mediated by the enzyme alpha-DAC1, which is alpha-tubulin acetyl transferase 1. This adds an acetyl group to alpha-tubulin, and HDAC6, which is a histone deacetylase that deacetylates microtubules. So what we've used are siRNAs targeting alpha-DAC1, where you can reduce alpha-DAC1 and you have a decrease in tubulin acetylation. And using tubacin, which is an inhibitor of the deacetylase, we can increase acetylated tubulin, as is shown here in the Western blots, as well as immunofluorescence images. So far, not much is really known about acetylation of microtubules in cell migration. But I'm just going to highlight what we've shown a few years ago, where astrocytes here again, which are migrating, we're looking at acetylation of microtubules and its effect on focal adhesion turnover in astrocytes. What we see is that upon depletion of alpha-TAT1, focal adhesions tend to have a longer lifetime. They assemble and mature and disassemble at a much slower rate as compared to the control cells. Also, when you have cells that are plated on rigid substrates and glass in this condition, alpha-TAT1 depleted cells migrate much slower, meaning that acetylation of microtubules promotes cell migration on these rigid substrates. So this leaves us with several open problems that I'll be addressing during this talk. What is the effect of rigidity sensing, and how do these cells, upon sensing rigidity, control the actin-myosin and microtubule cytoskeleton to then affect cell migration? So to start with, we plated astrocytes on soft and stiff hydrogels. And as you can see, with increasing rigidity of the substrate, starting from 1.26 to 48 kilopascal, cells have increasing levels of acetylated tubulin on stiffer substrates as compared to softer substrates. And as cells sense the rigidity of the extracellular environment through integrins, we decided to block a subset of integrins using RGD peptides, or use siRNAs targeting beta-1 integrin, which is a major mechanosensing integrin. What we observe is that tubulin acetylation decreases upon inactivation of integrins or blocking beta-1 integrin. So this suggests that rigidity sensing, along with integrin signaling, regulates acetylation of microtubules. What I did not show here is also, it's not just beta-1 integrin, but also the downstream signaling cascades, including SRC and FAC signaling that downregulates acetylation of microtubules. So acetylation of microtubules, like I said, is mediated by the enzyme alpha-TAT1. However, the regulation and functions of this protein remains unknown. So what we did was a mass spectrometry screen to identify potential interacting partners of alpha-TAT1. And one of the major hits that we found was talin, which is a major mechanosensing focal adhesion protein. And this interaction is confirmed here by.
Can I ask a question? What do you mean by a mass spec screen? You IP'd it and looked at what came to. Oh.
Yeah. So I have GFP alpha-TAT1 and GFP control cells.
So it's just an IP, basically.
Yeah, just a GFP IP, yes. We also confirmed this using GST pull downs here, as well as GFP IPs later on. Yeah. And so when we look at alpha-TAT1 in cells, by epifluorescence, you see alpha-TAT1 localizing to these microtubules. But what we also observed were these dashes or lines in these cells, which then made us look at cells by turf microscopy. And when we look on the bottom plane of these cells, what we see is that alpha-TAT1 not just associates with microtubules, but is seen at focal adhesions, as is shown here by vinculin positive focal adhesions, where alpha-TAT1 also localizes. And this localization, we then wondered whether the interaction of alpha-TAT1 and talin is important for this localization. So when you have talin depleted cells, what we observe is that alpha-TAT1 is lost from these, lost or minimal from these small adhesions that you see here, suggesting that the alpha-TAT1 talin interaction is important for this localization at adhesions. What I'm also not showing here is that when you deplete talin, the focal adhesion protein, you also have reduced levels of acetylated microtubules, and this is in line with its role in integrin signaling. All right, so alpha-TAT1 localizes to microtubules, like I said. So what happens if we get rid of microtubules? Here we use nocodazole to depolymerize microtubules, which also increases actin-myosin contractility. And what you see is that alpha-TAT1 is enriched at these focal adhesions. And when you when you target Rho kinase using a ROCK inhibitor, Y27, and you reduce actin-myosin contractility, what you observe is alpha-TAT1 is lost from these focal adhesions, suggesting that alpha-TAT1 localizes to these adhesions in an actin-myosin dependent manner. So what is the role of alpha-TAT1 at these focal adhesions? So first we wanted to look at what adhesions look like in these cells on soft and stiff substrates. And when you have wild-type astrocytes plated on soft substrates, you observe focal adhesions everywhere within the cell surface. Whereas on stiffer substrates, these adhesions are restricted to the cell periphery, as is quantified here. And when you deplete alpha-TAT1, what you observe is now this drastic change in the localization of focal adhesions, where cells now exhibit adhesions everywhere within the cell surface, similar to the phenotype that is observed in cells plated on softer substrates. On the contrary, when you increase acetylation using tubacin, cells on stiffer substrates still have adhesions at the cell periphery, and cells on softer substrates now have adhesions that are pushed or restricted to the cell periphery, mimicking the phenotype that you see on stiffer substrates. So just to summarize this bit here, when cells are plated on soft and stiff substrates, cells on softer substrates have less acetylated microtubules, and adhesions are well spread throughout the cell surface. And when you have less acetylated microtubules and cells are plated either on soft or stiff, they have this phenotype where adhesions are everywhere within the cell surface. And upon increasing acetylation of microtubules, cells behave like they are on stiffer substrates with adhesions at the cell periphery. So this means that acetylation alters mechanosensitivity of focal adhesions, but what does it do to the mechanical state of cells as a whole? So we then looked at the famous YAPTAZ mechanotransduction.
Can I ask another question?
Yes.
What's the evidence that it's only acting on tubulin? I mean, is anything else getting acetylated?
Alpha-TAT1, well, okay, so with tubacin, we do have other off targets, I think, because it's an inhibitor that inhibits HDAC6. Alpha-TAT1 is the only known acetylase of tubulin. So,
but the question is whether it also acetylates other things, like focal adhesion proteins.
Yeah, that's possible. We don't really know whether it acetylates proteins like tailin, for example. I don't think that's known. All right. So, yeah, we looked at the effects of acetylation of microtubules on YAPTAZ. And here, YAP is on stiffer substrates. It's been shown that YAP localizes to the nucleus. And on softer substrates, YAP is meant to be cytoplasmic, as you can see here in astrocytes as well. And when you deplete alpha-TAT1 with lower levels of acetylated microtubules, YAP is now in the cytoplasm, even on stiffer substrates. And again, on the contrary, when you increase acetylation using tubacin, YAP remains in the nucleus on stiffer substrates, but on softer substrates is where we see a striking difference with YAP translocating to the nucleus. This suggests that acetylation of microtubules regulates YAPTAZ shuttling and also the overall mechanical state of cells. So again, just a quick summary here, when you have cells with less acetylated microtubules, they behave like they are on soft substrates, and YAP is cytoplasmic. And when you have cells with higher levels of acetylated microtubules, YAP is in the nucleus, and cells behave like they are on stiffer substrates with higher tension. So we then looked at what happens to the focal adhesion-associated actin-myosin network. In migrating astrocytes, you have longitudinal fibers of actin that extend towards the cell front and are linked to adhesions at the cell front. You also have transverse arcs of actin that link cell-cell junctions and allow the monolayer of cells to migrate collectively. When these cells are migrating, you also have phosphomyosin light chain that localizes to the leading edge of cells. In alpha-TAT depleted cells, however, we have a striking phenotype where you lose these transverse arcs of actin linking cell-cell junctions, and longitudinal fibers of actin are pushed further back within the cell protrusion and do not reach the cell front. And this localization is also similar to phosphomyosin light chain, and adhesions are no longer at the leading edge of cells, but are again pushed within the cell protrusion. Along with this reorganization, we also wanted to look at myosin 2A. And what you see here, similar to phosphomyosin, is that myosin 2A does not reach the cell front in alpha-TAT depleted cells and is again within the cell protrusion, whereas in control cells, you nicely have myosin at the front as well as within the cell. So to look at this a bit more closely, we performed platinum replica electron microscopy, where cells are unroofed. Here, these are astrocytes that are unroofed. And looking closely at these structures, what you observe is focal adhesion associated actin bundles here at the leading edge of cells in control cells. These bundles also have microtubules and microtubules that go along these actin bundles, but also intermediate filaments that are wrapped around them, marked by yellow arrows here. In alpha-TAT depleted cells with lower levels of acetylated microtubules, what you observe is that similar to the immunofluorescence data, the actin bundles are pushed further back within the cell protrusion and are not at the cell periphery anymore. And these bundles are also much thinner as compared to control cells and do not have intermediate filaments wrapped around them. So this suggested that these thinner bundles, as well as changes in the reorganization of the actin-myosin network and focal adhesions, might regulate traction forces. So what we did to measure traction forces were plating astrocytes on micropattern hydrogels containing fluorescent beads, and upon tracking bead displacements, you can compute traction forces. So what you see here are stress maps of cells that are depleted of alpha-TAT1 or rescued using GFP alpha-TAT1. On the left here, alpha-TAT depleted cells with lower levels of acetylated tubulin have less tractions as compared to the control cells. And this phenotype can be rescued by overexpressing GFP alpha-TAT1 that increases traction forces. You can also increase traction forces by adding tubacin, which increases the levels of acetylation. And so all of this suggests that acetylation of microtubules promotes traction force generation. But how does this do so? So during cell migration, several Rho GTPases contribute to the different steps of cell migration, as is shown here. But I'm going to be focusing on one such widely studied Rho GTPase, that is Rho A. Rho A is activated through several GEFs, and I'm going to be talking about one GEF here called Rho GEF called GEF-H1. What's interesting about this Rho GEF is that it is a microtubule-bound GEF. So when it is when it's released in the cytoplasm and it's active, it triggers the Rho pathway to increase contractility. And when it's inactive and microtubule bound, you have less contractility in cells as it does not activate Rho A. So we then look at GEF-H1 in migrating astrocytes. In control cells, GEF-H1 is predominantly cytoplasmic, and you do see some speckles of GEF-H1 on microtubules here, suggesting that the dominant cytoplasmic pool of GEF-H1 shows that GEF-H1 here is in its active form. And when you deplete alpha-TAT1 or you have reduced acetylation of microtubules, GEF-H1 is decorating microtubules and are microtubule bound here, suggesting that GEF-H1 is inactive. And does this also translate to effects on Rho activity? So we did a Rho Techian pull down and looked at active Rho A. What we see is with alpha-TAT depleted cells, you have reduced levels of active Rho A as compared to control cells, suggesting that acetylation of microtubules promotes the release of GEF-H1 from microtubules to the cytoplasm, which then triggers RhoA, and which activates RhoA to control contractility. So how does all of this translate into a global phenotype? And so we looked at cell migration on collective cell migration and on substrates of different rigidities. And to do this, we developed an assay where we have, okay, so you're going to see a shadow of a microinjector here, needle with sodium hydroxide. So we're trying to create a local wound in a monolayer on hydrogels to study cells migrating to close this wound. So when you add a sodium hydroxide very locally to the monolayer of cells, you kill cells here, you wash them out, and you have cells that migrate towards this wound to close the gap. So as is described in several cell types, what we see is that astrocytes as well migrate faster on stiffer substrates as compared to softer substrates. And when you deplete alpha-DAT1, what we see is that cells on stiffer substrates are now migrating much slower as compared to the control cells. And this is similar to the phenotype that we observe on softer substrates, suggesting that acetylation of microtubules controls mechanosensitive cell migration. So just to conclude, and the working model that we're proposing is that when cells are sensing the rigidity of the extracellular environment through integrins, with the activation of integrins, you have signals that are sent back into the cell. Alpha-DAT1 is bound to tailin, and microtubules are acetylated. This means that GEF-H1 is released from acetylated microtubules into the cytoplasm to activate RhoA. This triggers actomyosin contractility, which then increases traction forces. And these cells migrate faster collectively. On the other hand, on softer substrates, with lower integrin engagement, cells have less acetylated microtubules. In this case, GEF-H1 is microtubule bound and is inactive, which does not trigger RhoA, so cells have low contractility and exert lower attractions and migrate much slower. So this kind of concludes the first part of my talk, but I'm going to be talking a tiny bit about what I'm currently up to during my postdoc. So I'm currently a postdoc in Professor Margaret Goddard's group at the University of Chicago. And here, what I'm trying to look at is mechanosensing via limb domain proteins in the context of vascular systems. So what we have here in normal arteries are endothelial cells that line the innermost part of these blood vessels. And upon narrowing of these blood vessels in atherosclerosis, due to the accumulation of lipids and calcium, what you have is a drastic remodeling of the endothelial lining within these blood vessels. And all of these rearrangements involves various mechanical cues that are sensed by these endothelial cells. And I'm looking at how mechanical cues are sensed here by a family of proteins called limb domain proteins. Some examples include zixin, paxillin, etc., that you're all probably familiar with. So several limb domain proteins are mechanosensitive, and here what you see is GFP zixin. When you laser ablate stress fibers, zixin is recruited to these stress fiber strain sites, and several other limb domain proteins such as FHL2 also undergo this similar process. So the questions that I'm trying to address during my postdoc are how do these limb domain proteins sense mechanical stresses here in vascular systems, and how do different mechanical cues sensed by these limb domain proteins transmit information? How do they transmit information within the endothelium, and how does that affect cellular functioning? All right, so I'm just going to thank all the people who have helped with this work, as well as my amazing mentors and colleagues, and funding and support from all these places. And thanks to especially Sandrine Etienne Manville, who is my PhD advisor, Margaret, who's my postdoc advisor. And if you're interested in reading more about my work during my PhD, the papers are here that you can have a look online. Thank you.
Thank you so much, Shailaja, for a wonderful talk. I'll start taking questions. I think there are quite a few. Starting with Rina's question, does talin and alpha TAT1 has direct interaction? Is it known that there is some binding?
We don't know whether it's a direct interaction just yet. We do kind of know which parts of alpha TAT1 and talin bind, but we didn't do any direct binding assays. We wanted to try bio-ID and other proximity assays as well, and we haven't gotten to that.
So thank you.
Great, fantastic. I just want to pop in with a quick sort of bookkeeping logistics note. We had a request to turn on the subtitles during the talk, which I think is great for accessibility, but you as the speaker can turn those off so that they're not distracting to you, and anyone in the audience can also turn those off as a Zoom setting if they're distracting or in the way for you. So just to let you guys know what happened there. Okay, so the second question we have is from Sang-Yoon Han, who says, great talk. Is it possible that the acetylation affects cells' own stiffness by stiffening microtubules and promoting more tension within F-actin? I think you touched on that a bit, but
yeah, possibly. We didn't actually look at cytoskeletal stiffness itself. We didn't probe the stiffness of the cell, but it does affect contractility and tension, so I presume that it would affect cytoskeletal stiffness. There's also a recent paper from Elias Barriga's group. I don't know if I'm pronouncing that right, but they do show that acetylation of microtubules regulates cell stiffness in neural crest cells. So that's something to look at, I guess. Yeah.
Cool. Next question is from Anna. She's asking, did you find KKN in your IP together with talin? Like, there are a few groups which has shown that it triggers a massive assembly of myosin 2A filaments, augmenting focal adhesions. Did you look at this sort of interaction in soft and stiff substrates?
I'm sorry, interaction with what again?
KKN.
Okay.
Anna, if you want to unmute yourself and ask that.
Sorry, yes, I was asking for KANK. So KANK is writing.
KANK, yes. KANK,
because Sasha group, they show that the interaction is related to adhesions, and then that recreates a massive filament formation of myosin and induce a growth of focal adhesions. But when you disrupt that,
we don't have KANK1. KANK2, I did do an IP, and I saw binding with KANK2, but it wasn't really on my mass spec, so I'm not sure.
Yeah, because Ben Gould, he has shown all these interactions with KANK, and that is the way that they have been explaining the role of microtubules in focal adhesions.
Yeah, I mean, we do have some data to show that KANK and acetylation of microtubules are, KANK regulates acetylation of microtubules, but I don't really know if they interact.
Yeah, that was my question, if the acetylation of microtubules has something to do with KANK, if that preserves that there or not.
Yes, so when you deplete KANK1 and 2, you have an increased level of contractility, as Sasha Barshetsky had shown in their paper, and this increases acetylation of microtubules as well. Yeah.
Okay, thank you.
All right, we do have a few more questions. In the interest of time, we might have to return to these afterwards, but I'll ask one more from Ernest Liu, asking, how does the alpha TAT1 sense the substrate stiffness?
I wouldn't say that alpha TAT1 itself senses substrate stiffness. I think it's through integrins and adhesions that sense the substrate stiffness, which then affects alpha TAT1. That answers the question very quickly.
Okay, let's just do, I don't know. I want to squeeze in one more and then we'll move on. So from Michael Kingprama says, excellent talk. Have you also checked expression downstream targets of YAP-TAZ signaling when you knock down the alpha-TAT1 and the astrocytes using the soft and stiff substrates?
Yeah, that's a great question. No, we haven't looked at downstream. Yeah, that was something that I was planning to do, but I didn't get to it. But yeah, I think it would be important to look at downstream targets of YAP-TAZ.
Shailaja, there is a question from Sophie. Maybe you can like answer it in chat.
Yeah, I can do that. Okay.
Thank you. Thank you so much.
Thanks so much. That was really fantastic. We're going to switch gears. Joseph, if you want to start sharing your screen while I introduce you, and you may want to look for the setting that lets you remove the subtitles so that you can't see them, so you can hide them. Okay, great. So we are so excited to welcome our second speaker today. We have Joseph D'Alessandro. There are a number of French names in his bio, so I apologize for mispronouncing those. So Joseph is currently a permanent researcher at the CRNS in in the lab of Benoit Ledoux. And he is focusing on the relationships between short-scale cell-cell and cell-substrate interactions and large-scale properties of movements within tissues. He uses in vitro microfabricated environments and data-driven active matter modeling. So I'm sure we'll be hearing more about all of that today. And he has also worked previously for his PhD in looking at long and short-range interactions and cell migration in dictyostelium. I'm trying to do the talk backwards here, and I don't know if it's working super well. So anyway, he has lots of experience in physics and biology, and really excited to hear what he has today. Thanks so much, Joseph. Take it away.
Thank you, Jennifer, for the introduction, and thank you for this opportunity to reach this such a broad audience worldwide from the lab. It's exciting. So today I'll be talking about this work that I've been doing those last few years in this lab, the lab of Benoit Ledoux and René-Marc Meige in Paris, about how cells might store some kind of information in their environment. So this, what I call this external memory during their migration. So this talk will be much less molecular and much more about trajectories of cells. And so I'm mostly focused in seeing how we can read cell trajectories to kind of infer what happens within the cells. And so as we might know, cell trajectories reflect kind of the dynamics that happen intracellularly, especially the dynamics of the polarity of the cytoskeleton. But they also reflect the cell-cell interactions or the interactions that cells have with their substrate. And so the common paradigm for cell migration is that single cells move through random walks with kind of short persistence, which makes them kind of difficult to study when we look at trajectories of cells in 2 dimensions. So what I will present today starts from trying to simplify the system by plating cells on basically lines, so one-dimensional lines of fibronectin that we print on the substrate, and where we can see that we placed isolated MDCK cells, and then we looked at how they would behave on such a system. So when we do so, here is the result. So we get many cells moving on their lines, and we can follow them in time. So you can see here that I've pointed the different single cells that we see, and we can follow them in time. So what we can usually do is to take all those trajectories, measure their properties, put them in kind of bags, and look at the distributions and see what we can infer from that. But another thing that we can do is just to zoom in and look more closely to single trajectories and see whether we can see some specific behaviors. And so what happens when we look at one single cell moving in time is that it really appears to have a very strange way of moving. And so if I show the corresponding kymograph of this cell, so just basically snapshots of the cell that are put together in time, you see that this cell is strikingly oscillating around its initial position. And so when we do so, we want to wonder whether it actually relies on real migration. And so to do so, what I've done was to look at this reporter of the cell polarity, which is the so-called PBD YFP reporter of activation of RAC1 and CDC42. And so we can see that when the cell is moving, it's actually polarized, and it's really polarized during long runs, but then it seems to turn back at very constant locations in time, and it kind of abruptly turns back. So what happens there? First, if we look at many trajectories, we see that they don't all look exactly the same. So all those curves are kymographs of different single cells in the same conditions. We see that they're a bit different from each other, but still we see that they have like common features, and mostly they almost all oscillate in time, and they almost all oscillate around their initial position. And so the first question is, how can we extract common features from those various resembling but different trajectories? So the first thing that we can do is, sorry, is to analyze the oscillations of the cells. So if we analyze the oscillations and we compute, for instance, the amplitude and the time period of those oscillations, what we can see on those two examples is that first, the amplitude of the oscillations scales almost linearly with the time period of the oscillations, which kind of says that the cells are moving at constant speed, even though the time period is not constant in time. And also what is interesting is that if we look at the time series of the oscillations, we see that the amplitude is slowly growing in time, which shows that there is a consistent increase of the period and the amplitude of the oscillations in time. And if we look at many trajectories again, so again, all plots are for a single cell, we see that this relationship between amplitude and period of oscillation is kind of kept for all single cells. So all single cells seem to move at a roughly constant speed in time. And again, if we look at many different single cells, we see that the amplitude of the oscillations is increasing slowly in time. So that is very striking. And then we can wonder why cells are behaving in that way. So the hypothesis that we had is that to explain that cells are first oscillating and then they're increasing the amplitude of their oscillations in time, is that the cells might be able to deposit a footprint on their path, on the substrate. And once they reach the edge of this footprint, they prefer to turn back on their footprint rather than persisting in the same direction of motion. And since at each oscillation, they're able to overshoot a little bit from their previous location, they might be able to extend this kind of footprint that they make on the substrate. The alternative hypothesis would be that the cells have a kind of intracellular clock that would lead to those oscillations, but then it would need to be very synchronized with the motion itself to allow for this decreasing frequency or increasing amplitude of the oscillation in time. So to check this hypothesis, we resorted to what we call conditioning of the substrate. So to do so, what I did was to first prepare substrate, so still fibronectin lines patterned on the substrate, then first seed cell at very high density so that they cover all the available adherent surface. And then if they're able to deposit any footprint on their way, we imagine that when the substrate is confluent, then the conditioning, the footprint will be completely covering all the fibronectin patterns. Then we would detach the cells so that we leave substrates without cells, but with this cellular footprint on the substrate. And the expectation is that if we plate again isolated cells on such substrates, then the cells would be able to move freely without encountering an edge of their own footprint. So what happens when we do so? If we compare, so on top it's a control cell, so a cell on a normal fibronectin pattern, and those cells on conditioned substrates, what we see is that the cell on conditioned substrates is no more oscillating, but it's moving super persistently on the line, almost as if there were like no turning back, except probably at the end of the line here on the right, at the beginning of the movie. So if we turn to camera graphs again, so corresponding to the movies you've just seen, we see that the control cells that really oscillate with increasing amplitude, while the cells on conditioned substrate, it's able to move very persistently up to very, very large distances that we're not really used to see in such experimental setups. And so that really probably means that cells are like really able to deposit such a footprint, and they're then able to move persistently within that footprint. So another way to show it is if we look at many different trajectories on those same conditions, we see that cells on control conditions, so here in blue, they're moving very, very like close to their initial positions, while the cells on conditioned substrates, they're moving really, really far away from their initial position. So that leads to this idea of kind of self-attracting walk, where the trajectory of the cells is attracting the cell itself to previous positions. And so to better understand what it means for the exploration of space, we resorted to modeling and simulations in collaboration with Raphaël Voiturier and his PhD student, Alex. And so what they did was to design a very simple model where agents, so a cell, let's say, is able to jump on a discrete network from one side to the other. But then there is just two simple rules. When the agent is within the known kind of footprint, it moves with some kind of persistence. But when it reaches the edge of the footprint, the probability to turn back to the already known position is much higher than the probability to discover a new site. And so as we see here on simulated trajectories, very long time trajectories, the behavior can be qualitatively similar, and the rules that we entered are qualitatively similar to what the cells are doing. But the question is, what could we learn from this kind of approach? So first, to learn anything from that, we need to be able to compare the experimental trajectories with the simulations. So at the beginning, it does not really look very similar. But then what we tried to do was to discretize the experimental trajectories. So first, passing from continuous space to discrete space, and then discretizing again in time, so that from a real trajectory, we end up having like a trajectory that really looks like the trajectory of the agent in the simulations, where you only have a cell jumping from side to side on each substrate. And what's interesting from that is that we can first compare the experimental trajectories to the simulated ones, but we can also kind of infer the parameters that we might input in our modeling in order to recover the similar behavior in the simulations. So this model has only two parameters. So the first one characterizes the free persistence of the motion. So basically, it's the persistence of a cell that will be on a homogeneous footprinted kind of substrate. And the second parameter is the strength of the attraction of the footprint, meaning that when it's on the edge of the footprint, the higher the beta value, like the more negative the beta value, the more the cell will have a tendency to go back and not to go further away on the not yet known substrate. And so the first thing that we can do is to kind of measure those parameters on single trajectories. So on those plots, every single point corresponds to a single trajectory. And what we can see is that there is, of course, a variability between the cells, but all the points that we get from all these different trajectories, they all fall on a single curve. And if we compute the probability to come back on the footprint when the cell is on the edge, what we find is that strikingly, this curve where all the points fall on correspond to the fact that all the cells, even though they might have different propensities to be persistent when they're on the footprint, when they arrive on the edge, they all have kind of the same probability to go back. So that points to some very common behavior of all the cells in that very varied population. Then, of course, the effect of conditioning will be to decrease the absolute value of beta, so decrease the attraction of the footprint, meaning that basically cells don't see any their own footprint because all the substrate is covered with the footprints of all the cells that have been there before. Then what's interesting with that modeling approach is that it allows to make predictions, and predictions that can be tested in our experiments. And so I would like to just insist on three main properties that this model underlines. So the first is that trajectories kind of age in time. Then the second one is that cells go less far, but they should be able to explore better the space around them. And the last one is that we can also make predictions of what happens in 2D, as at the moment, everything has been done in one dimension. So first, aging of the trajectories, what does it mean? So basically, if we compute, so here it's basically the mean square displacement. So it's a measure of how far the cells are going from their initial position. What the simulation, what the model predicts is that this measure should age, meaning that when you compute those MSDs at different timings in the experiment, you should not find the same result. And it's actually what we find. Basically, when you compute the displacements of the cells at early times, the cell is not going very far away from its initial position. But when you compute it much later, then the cell is able to go much further away because it has discovered much space already. So it's able to go further, meaning that the measurements that we do, they really depend on time. And so that's super important because experimentally, when we do measurements of displacements, if we wait like, let's say 12 hours before we place the cells into the microscope, we won't get the same result as if we measure the displacements right after having seeded the cells on the substrate. And this effect, of course, disappears when the cells are on conditioned substrate, because this aging is really a feature that comes from the footprint that we don't see anymore on those conditioned substrates. Then what happens in 2 dimensions? So I mentioned that at the very beginning of the talk, that trajectories in 2 dimensions, they are quite complicated. They resemble to, let's say, random walks, but they have very strange properties. And the thing is, because cells are very variable from one cell to the other, and also because the persistence is very short as compared to the scale of the cell. So it's a bit difficult to really assess what are the rules leading to the trajectories that we find. And especially if there are subtle effects in the motion of the cells, they might be difficult to see just looking at the 2-dimensional trajectories. So basically, if we look at trajectories now of cells on control substrates and conditioned substrates in 1D, so I recall what we've seen before, the cells are oscillating on control substrates, and they're not going very far away from their initial position, while on conditioned substrates, they move very persistently up to very large distances. And actually, the same happens in 2 dimensions. So when we now play the same cells, but on 2-dimensional substrates, so just fibronectin plated all over the surface, we see that cells on control substrates, so just raw fibronectin on the substrate, the cells are moving around their initial position, but they're not going super far away from their initial position. While when we first condition the substrates, meaning that we put first a confluent layer of cells that we wait for incubation for some hours, and then we remove the cell and we place again single cells, we see that the cells are able to go much further away. So the same effect appears again. And what is more, maybe, the model predicts another interesting feature. So again, we see aging in 2 dimensions, meaning that, again, the mean square displacement, the measure of this mean square displacement depends on the timing of the measurement. So if we measure at the very beginning of the experiment or the simulation, we'll get small mean square displacements. Basically, the cell is not able to go very far away. But if we measure those MSDs at very late time points, then we see that the cells are able to go much further. And this disappears on conditioned substrates. But what is more, at long time, we see this kind of slope here in the plots of the MSD. And what it tells, this slope, so you see it's different in conditioned substrates and in control substrates. Basically, what it tells is that we have subdiffusive motion. So in conditioned substrates, we have diffusive, like the normal expected motion for a random walk on 2 dimensions. We have normal diffusive motion. But on control substrates, where the cells are experiencing this footprint, which is attracting themselves to their previous positions, we see that there is subdiffusion. And basically what it means for single trajectories. So the first thing, of course, as we've seen before, is that on control substrates, so all those are single trajectories that I've put on a grid for readability. So first, cells on control substrates, they move not very far away from their initial position. And second, more qualitatively, but maybe more importantly, the cells on control substrates, they're really roaming around their initial position. And so all their positions, they're really well covering all the space around their initial position. So you see the initial position is here, and then the cell is really like turning all around its initial position. While in the conditioned substrates, where the cells are like kind of moving in a more diffusive way, they are going further away, of course, but still they are not exploring all the space around their initial position, which means, for instance, that if the cell is looking for a target that is located just nearby, in that case, it's not going very far away, but it's able to find the target all the time because it's going all the time all around its initial position. While here, it could go very, very far away, but it could also miss a target that would be located very close from the initial position just because it's escaping super fast from its position and it's not really exploring the space that's all around. So in the end, what we've seen is that we have more deterministic patterns of motions in 1 dimension, which allowed us to detect this effect of conditioning and of footprint deposition, which is very subtle and it's hardly seen in 2D. So that shows that those kind of model experiments and predictions are important to decipher sometimes subtle effects in the migration of cells. Also, we've seen that the similar free persistence of cells leads to the hypothesis that probably the cells that they kind of optimize their substrate, so that they have a kind of template over which they print whatever is necessary for them to move, and maybe they're optimizing it. They're able to build themselves their best environment to move. Then this aging effect is important because then it means that when we are running cell migration experiments, the timing at which we do the measurements is super important if we want to do quantitative measurements. And then in more qualitative meaning is that the cells escape slower from their initial position, but they explore better the space around them. So the questions, of course, that it leaves behind is that we don't really know what this footprint is. So we've done some immunostainings first, and we've seen that fibronectin from the cell and laminin are deposited by the cells. So this is a decellularized condition pattern. So you can see the patterns here, and you can see that fibronectin and laminin are left behind by the cells. That was also quantified in those plots. But of course, that doesn't mean that they only leave fibronectin and laminin. That cells can leave like many things on their way. So that's still to be discovered what the footprint is really made of. And another interesting feature that I wanted to share with you is that in our hands, at least, the density, the surface density of the fibronectin that we put initially on the lines is actually not very important for the properties of migration of the cells. So basically, they all behave the same, whatever the surface density of fibronectin that we have. And you see that we have a quite large range of concentrations of fibronectin that we have covered. So which means that for them, the patterns that we make, so the substrate that we give to the cell is just a kind of template that then they will optimize by printing whatever is important for them to move. Yes, as I have mentioned, the chemical nature of this footprint is still to be discovered or to be completely characterized. What it means is that the condition substrate that the cells made is kind of optimized for their migration, and it I've not shown it, but it allows a better mechanical coupling between the cell and the substrate. The cells are able to pull more on the conditioned substrates than when they are on the edge of their footprint, so on non-footprinted, let's say, substrates. And of course, the last question that it leaves behind and that we would like to answer now is, what is the reason for a cell to turn back when it reaches the edge of its own footprint? And that would be a very exciting, I think, question that I'd like to address in the future. So with that, I'd like to acknowledge the people that who participated in that work. So people from my lab in Institut Jacques Monod in Paris, and also the people are theoretical collaborators, and especially Alex, who did most of the work on the simulations and the modeling. So all this work is now published in Nature Communication, so you might want to read more in there. And now I'd like to thank you all for your attention, and I'm very happy to be able to answer questions, if any.
Wonderful, thank you. That was fantastically interesting. I have some questions, but we'll start with ones from the audience. From Emiliano Perez Epina, as asking, what if instead of the cells depositing a footprint, what if they're just aligning the ECM that's already there?
Yeah, that's a great question. So, and we also asked it. So at least at the scale that we're able to explore, like, let's say with confocal microscopy, we don't see any change in the structure of the fibronectin that we plate. So basically the fibronectin that we put on a substrate, it's kind of globular, it's super homogeneous on the surface. And if we look at the same after conditioning or after having putting cells on the way, it still looks super homogeneous. While if we look at fibronectin that we had in the medium during the cell migration, or if we look at the fibronectin that is produced by the cells themselves, all those fibronectins are differently structured. I might have an image. If I find it quickly, I can show. No, I don't have it, sorry.
Thank you. The next question is from Abhishek. So he has two different questions. So I'm going to take the first one. Physiologically speaking, at what level receptors or any downstream signaling do you think this footprint information is imprinted in the cells?
I think it's really a matter of like, if you mean by that, the question is about how it's kind of sensed by the cell. I think it's
so it's more at what level do you think this information is coming from, whether it's at the receptor or something downstream of that?
No, I think it's really a matter of cell substrate interaction. So really at the level of the focal adhesions probably. So as I've mentioned, the cell substrate coupling, so in terms of at least of traction forces, but also in terms of the mechanics of the cell itself, it changes when the cell is on the edge of the footprint, as we have characterized. The cell kind of seems to soften when it's on the edge of the footprint. So I think it's really sensing a different, probably adhesive environment when it's reaching the edge of the footprint. So I think it's really.
But that could be still with receptors, right? Like that's probably where the interaction is happening.
Yeah, it's true. Yeah, it's like through adhesion. So probably through integrin receptors or
the second part, the second part of his question is also about cell-to-cell variability. So he's asking the beta value that you observe could be different for different kinds of cells. Do you have something to sort of comment on that? Like, did you look at various kinds of cells too?
So I have looked at different kinds of cells. Truth is, I've not measured this beta for the different cell type that we've studied. So the cells that reproduce best the behavior of those MDCK cells were KCO2 cells, so other epithelial cells that form monolayer for what it means, I don't know. But some cells are able to oscillate more or less, and some cells, I think, are able to overcome this footprint effect. So that's another question. But yes, so to answer precisely the question of this beta value, I don't have the answer yet, but at least what it means for MDCK cells, what was super interesting, I think it was not the beta value itself, but like the relationship between the two parameters that really shows that despite the variability between the cells, they all have the same probability to go back when they're on the edge. So that was the most striking fact.
Okay, fantastic. We have two questions from Itaka Matubayashi. So he says that MDCK cells are famous for being able to be induced to undergo EMT by HGF. And so he's wondering if in your system you see any alteration of behavior with HGF, if you've tried that.
No, I have not. It's one of the, it's true. It's one of the many things that we could do to now kind of screen, because one of the idea now is kind of screen in what conditions cells do behave in such a way. So one of the screen I was envisioning was just to screen different cell types, but indeed trying to tune the behavior of this single cell type. So for instance, using AGF would be amazing, I think. Yeah, I've not thought of this, but yeah, good idea.
Okay, great. And then the second part of his question, which is also relating to what I was thinking about. So MDCKs, of course, we normally see them as, collectives, right? They undergo collective migration when they're crowded and stay epithelial. And so have you looked at how you can see a group of cells that are adhered to each other on those tracks and how they behave?
So I've not really analyzed it yet, but I have some data, and of course I'm super interested in the behaviors that it will imply. So one of the first thing that I can answer at least qualitatively is that it's kind of funny. We don't see really well, but I can like just describe it. In this precise example, you see the cell is oscillating, so probably within its own footprint, but at some point, there are cells coming in and those cells are coming, are like escaping again. But then what we don't see here, because I've just cut the chymograph, is that now this cell is able to follow on the track, and it's not touching the other cells. So it's really still moving by itself, but it's now able to go and to follow. So of course, I believe that we have amazing collective effects that happen with that kind of footprint deposition. And indeed, I expect that it will change a lot the properties of the group motion if we played groups on conditioned or not conditioned substrates.
Yeah, I think there could be really profound effects of in groups. That's really interesting.
Yeah. And especially to go further, we usually, I mean, there are examples of studies that look at expansion of cell colonies using MDCK cells, but they're usually not really moving, I mean, as a whole. And I expect that maybe on conditioned substrate, in small groups at least, they might be able to move as a whole, because single cells, they really move like crazy compared to the ones on controlled substrates.
Mm-hmm. We have another question from Emiliano saying it would be expected that the oscillations happen in 2D as well, unless there is some diffusion of the deposit footprint that is happening. Do you have any thought about it?
So I was expecting the same, I have to say. But actually, discussing with our collaborators, it's not really expected from the theory. So because oscillations is an outcome in 1D, because it's the only way to come back within the footprint when the cell is on the edge. But in 2D, it's not mandatory to oscillate. The cell might just turn back into the footprint without really oscillating, and that's actually the dynamics that we see. So I'm trying to go back to this trajectory. So you see, the cell is more like turning around its initial position, but it's not really oscillating, because it can also turn within the footprint. So it leads to a motion that it's not really oscillating, but it's still sensing the edge of the footprint.
Okay, great. So Giorgio is asking, you've addressed this a little bit, but look about sort of different cell types. So specifically, have you tried conditioning the lines with different cells that are specifically displaying different frequencies and amplitudes? And then how does this conditioning behavior then affect the migratory behavior?
So I've definitely not done it in a very exhaustive way. I'm wondering, I've tried to do kind of cross conditioning experiments. I think it definitely works. I'm trying to think. I might have checked for MDCK and KCO2, if we condition with MDCK cells, then KCO2 cells will be able to sense this footprint from the MDCK cells. So this, for sure, is an effect, but no, I've not really explored that much further, how it relates.
Thank you. I think we will take the last question before we wrap up, from Marco Antonacchi. Wondering, I think it's a more technical question, but wondering which method do you use to remove the first layer of cells for the footprint? And if you have tried multiple methods to do that.
Yeah, I've tried two basically. So the first one was trypsin, which is very bad, as you can expect, because then it removes all the proteins that are on the surface. So when I use trypsin, then cells just barely attach when I add cells afterwards. And then I tried the EDTA only, and it works fine. The only drawback is that the detachment takes a long time, 1 to 2 hours. But yeah, that is the way I detach cells. So that I only detach the cells, and I'm trying to detach smoothly the cells so that the surface is not hurt.
Thank you so much. I think with this, we will wrap it up. Thank you so much, Joseph and Shailaja, both for giving such wonderful talks and kickstarting our series for this season. And we all hope to see you next Tuesday, I guess. Thank you so much.
Thank you.
Thanks,
you.
everyone. See you next week.